A myofibril contains units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving a skeletal muscle its striated look. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M-line. Thin filaments are composed of the protein actin, which are anchored to the Z-lines on the outer edge of the sarcomere. Because the actin filaments are anchored to the Z-line, sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of the muscle contraction. Sarcomeres shorten without thick or thin filaments change in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing the myosin to pull the actin filament towards the M-line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to the actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding or more contraction, or remaining unattached to allow the muscle to relax. The muscle contractions are controlled by the actions of calcium. The thin filaments are associated with regulatory proteins called troponin and tropomyosin. When the muscle is relaxed, tropomyosin blocks the cross-bridge binding site on actin. When calcium ion levels are high enough, the ATP is present. Calcium ions bind to the troponin, which displaces the tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to the binding site on actin, forming a cross-bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium storage. Calcium ions flow to the myofibrils where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z-line draws closer to the M-line. As the sarcomere in myofibrils contract, the entire muscle fiber will shorten. When the muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to move freely. The term neuromuscular junction refers to the synapse between a motor neuron and a skeletal muscle fiber. When a motor neuron approaches a muscle, it branches out into several extensions that end in areas called axon terminals, which can release neurotransmitters. These axon terminals are situated over a specialized region of the muscle called the end plate. The axon terminals are separated from the end plate by a space called the synaptic cleft. The end plate beneath each axon terminal contains several deep indentations called junctional folds. These junctional folds contain high numbers of ligand-gated ion channel receptors for the neurotransmitter acetylcholine. When an action potential travels down the motor neuron, it causes the release of acetylcholine into the synaptic cleft. Acetylcholine binds to acetylcholine receptors in the junctional folds of the muscle membrane, which causes ion channels to open to allow positive sodium ions to flow into the postsynaptic cell. This produces a depolarization of the cell called an excitatory postsynaptic potential, also known as the end plate potential when it occurs at the neuromuscular junction. This depolarization leads to the opening voltage gated sodium channels, which causes the end plate potential to lead to an action potential. This action potential travels along the muscle fiber and causes contraction of the muscle. The enzyme acetylcholine esterase is also present at the neuromuscular junction. It breaks down acetylcholine and in the process terminates its effect on the muscle fiber, thus ending the communication between the motor neuron and the muscle fiber. Sliding filament theory is the explanation for how muscles contract to produce force. Actin and myosin filaments within the sarcomeres of muscle fibers bind to create cross bridges and slide past one another, creating contraction. Myosin has ADP and phosphate attached to the individual myosin heads. Actin has tropomyosin and troponin. When we move, calcium is released from the sarcoplasmic reticulum and connects to the troponin molecules, causing the tropomyosin molecules to move. The single phosphates move so the myosin can connect with the actin. This is called a cross bridge. then remaining a power stroke when the myosin heads pull the actin inwards. ATP connects to the myosin heads, which trigger the release of the myosin heads from the actin attachment sites and are broken down into ADP and phosphate as the heads fall back into resting position once again, which is called the recovery stroke. If calcium is still present, the cycle will repeat again and the sarcomere will continue to shorten until the calcium ions are transported back to the sarcoplasmic reticulum and the muscle relaxes. Energy is provided by the breakdown of ATP by the class of enzyme ATPase. In order for the muscle to contract isotonically, the muscle must receive an instruction to do so. In order for the instruction to be carried out, there needs to be a sufficient stimulation from the nervous system. The nervous system sends impulses to generate an action potential. With sufficient stimulation, the neurotransmitter acetylcholine is released, causing the release of calcium. Eyes detect the stimulus. The sensory organs are the eyes in this case. Info comes in through the sensory organs to the brain. The brain makes a decision, sends an impulse down to the spinal cord. Impulse through the motor neurons to the muscles we want to contract, like the quadriceps extends to the knee. Motor units are made from a single motor neuron and all the muscle fibers affect it, motor neuron branches. Finally, onto the types of muscle contraction. A muscle fiber generates tension through actin and myosin. While under tension, the muscle will either lengthen or shorten, or remain the same. Although the term contraction implies shortening, when referring to the muscular system, it means the generation of tension built up in a muscle fiber. Several types of muscle contraction occur and are defined by the changes in the length of the muscle during contraction. An isotonic contraction is a muscular contraction in which the length of a muscle changes. Isotonic contractions will remain constant tension in the muscle as the muscle length changes. They can be either concentric or eccentric. An isometric contraction is a muscular contraction in which the length of the muscle does not change. In contrast to isotonic contractions, isometric contractions generate force without changing the length of the muscle. An example would be the muscles of the hand and forearm responsible for grip. Using this example, the muscle contraction required to grip but not move a heavy object prior to lifting would be isometric. Isometric contractions are frequently used to maintain posture. Isometric contractions are sometimes described as yielding or overcoming. An eccentric muscle contraction is an isotonic contraction where the muscle lengthens. An eccentric contraction results in the elongation of the muscle while the muscle is still generating force. In effect, resistance is greater than force generated. Eccentric contractions can be both voluntary and involuntary. For example, a voluntary eccentric contraction would be the controlled lowering of a heavy weight raised during the above concentric contraction, or a bicep curl. An involuntary eccentric contraction may occur when a weight is too great for a muscle to bear, so it is slowly lowered while under tension. Cross-bridge cycling occurs even though the sarcomere muscle fiber and the muscle are lengthening, controlling the extension of the muscle. Finally, concentric. It's an isotonic contraction where the muscle shortens. A concentric contraction is a type of muscle contraction in which the muscles shorten while generating force, overcoming resistance. For example, when lifting a heavy weight, doing a bicep curl, a concentric contraction of the biceps would cause the arm to bend at the elbow, lifting the weight towards the shoulder. Cross-bridge cycling occurs, shortening the sarcomere, muscle fibers, and muscle.